so let's take a look at the structure of DNA. And we're starting off with this guy named Phoebus Levine, and this was in 1929. And he determined DNA's main parts, so the main parts of DNA, not its overall shape and structure. So he found that DNA is made of nucleotides. So this is showing one nucleotide, and each nucleotide has 3 parts to it. So each one is made of a phosphate group, shown here, a deoxyribose sugar, shown here, and a nitrogenous base, right here. And there are 4 different nitrogenous bases. So if we go back to this previous picture, 2 of these nitrogenous bases are called purines. So adenine here is a purine, and guanine is a purine. So this is the nitrogenous base, which makes that a different nucleotide. All right. Next are pyrimidines. So thymine and cytosine are pyrimidines. All right. And all I want you to note about this is that an adenine and guanine are larger than the cytosine and thymine. All right. So let's move on to the next guy here. And so this is Char Graph. And this was in 1950. And so what Char Graph was looking at is he was looking at the percentages of adenine, guanine, cytosine, and thymine, or A, G, C, and T, in different species. And so he hypothesized that they were, 25% each. But what he found was that these percentages were different in each species. All right? But there were certain things that he always found. All right? So what he always found here was that his percentage of adenine always equaled his percentage of thymine, and his percentage of guanine always equaled his percentage of cytosine. So if you look on here, here's adenine, there's thymine, guanine, cytosine. Not exact, but really, really close. All right. And also what he found is if he took his, if he added up his adenine and guanine, well, that equaled 50%. And if he added up cytosine and thymine, well, that equaled up 50% as well. So he came up with what is known as Char Graph's rules. In that state, that the amount of adenine, guanine, cytosine, and thymine in DNA varies from species to species. But in each species, the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. All right, let's look at the next picture, or the next person here, which is Rosalind Franklin. And this was in 1951. Now, Rosalind Franklin, with a few other people, were in a race to determine what the overall shape and structure of DNA was. So it was, Hershey and Chase's study didn't come out until 1952, but as I mentioned earlier, there were other studies in the late 1940s that were suggesting that DNA was genetic material. So then there was a race on to figure out what the overall shape and structure of DNA was. All right, because they thought whoever could figure that out would win a Nobel Prize. All right, so Rosalind Franklin was part of that race, and she did this method known as X-ray chromophotography. And in X-ray chromophotography, you take some X-rays and you shoot it at a molecule, and you put some photographic film behind them. And based upon the shape of that film, they could determine what the shape of the molecule was. And realistically, pharmaceutical companies still do that today. Now, this is one of the pictures that she took of DNA. And from that, she was able to determine that DNA has a shape, has a helix shape. All right? So, it wasn't until the next guys, using Rosalind's picture here, that they were able to determine the overall shape and structure of DNA. And so these two guys are James Watson here and Francis Crick here, and this was in 1953. So they determined the overall shape and structure of DNA, and as I mentioned, they used this picture of Rosalind Franklin's. Now, this was unbeknownst to her. A colleague of hers had taken this picture out of her possession and brought it to these guys, and they used that and were able to determine that it wasn't just a single helix, but it was a double helix. All right? So, they're standing next to a model of this here. And so this is a nice little diagram of DNA here. Here's this computer-generated model of it. All right? But here's what we see. So it essentially, it's like a ladder that you've twisted. All right? So it makes this winding staircase, another way to look at it here. But if you untwist that ladder, this is what it looks like here. All right, and so what we see on the sides here, or the sides of the ladder, is we have this phosphate sugar backbone. So, we see that there. And then the nitrogenous bases are directed inward, all right? So this is one strand on this side. Here's the second strand on the other side, so double stranded, all right? But there's a particular pattern that shows up here in that adenine bonds by hydrogen bonds to thymine on the other strand. Cytosine on one strand binds to guanine on the other strand. So you can see those hydrogen bonds between those. This kind of makes sense based upon what we found out from Char Graph, is that his amount of adenine equals his amount of thymine, and his amount of guanine always equals his amount of cytosine. So you can see that in the molecule itself on how these different strands bind to each other. This is known as complementary base pairing. A with T, G with C.